Okay, dah klik tapi dia tengah loading lagi.
Okay. Maybe is your internet okay?
Okay dah.
Okay. So try to look at your YouTube. Because
Okay.
OBS dia dah start streaming dah, so it should be okay lah.
Okay, yeah.
Okay, can you see the
Is
room?
there music?
There's no music because aku minta music tu tak dapat lagi. Ke you want me to use the same music yang hari tu saya letak? What is that? I couldn't hear you. Okay, kalau aku letak lagu yang hari tu?
Boleh je.
Alright.
Anything lah.
Okay. Should it? So, the VIP have entered?
Belum.
Belum Okay, no problem. Media source. Alright.
And then got the benda dan running thing, right? Apparently the ones that we are talking now also they can hear in YouTube itself.
I don't think so because I didn't put audio output for the intro. The microphone. So right now it's intro background, right? So
Yeah, but then they can still hear what we're saying in the YouTube.
Oh, can they?
Sekejap.
Still boleh dengar.
Still boleh dengar.
Do you want me to start the slides straight away at 10?
No, doctor. I will give an introduction first.
Okay, all right.
All right. So, can I start? Hold on. All right. Hello and good morning. I bid to all. I am Hashwin Ravichandran, a second-year genetics student from UKM. I will be the moderator for this webinar. First of all, allow me to explain the flow of the event for today. We shall begin with a talk given by our guest speaker, followed by a Q&A session. Feel free to type out your questions while the talk is being given in the comment section at the side. These questions will be collected and conveyed to the speaker during the Q&A session. And be sure to wait until the end of the webinar, as a QR code will be displayed for your attendance and feedback. Only those who have filled out this form will receive the e-certs. So be sure to wait to get the link and to scan the QR code at the end. All right. So moving on to the first segment of the day. With me today, we have our esteemed guest, who is a professional from the genetics field itself. She is none other than Dr. Arshana Amin. Dr. Arshana is currently a research and laboratory manager at the Malaysian Genomics Resource Centre. She completed her bachelor's in biotechnology at IIUM before pursuing both her master's and PhD in biosystem sustainability at Hokkaido University, Japan. While studying, she also managed to publish several publications on various gene and disease-related studies. Apart from that, she has also been involved in many projects together with government and private agencies such as the Malaysian Cocoa Board, Felda Wellness Corporation, Pure Circle, and many others. Without further ado, I shall open the platform to Dr. Arshana to give her talk on the very much anticipated topic on a career in genomics.
Okay, thank you so much for the introduction, Harshwin. So I will start with my presentation this morning. Okay. So my name is Arshana, and I'm currently the Research and Laboratory Manager at Malaysian Genomics Resource Centre, Berhad. So today I'll be talking a little bit about our company and what we do and what it takes to be in this field. Since there will be separate Q&A session at the end of the talk, so feel free to jot down your questions so that we can discuss them at the end of this talk. So as a brief introduction about the company, Malaysian Genomics Resource Centre is a service provider that provides lab services such as sequencing, bioinformatics analysis, genetic screening, and CAR T cell therapies. We also provide other services such as product distribution, project management, contract research, and bespoke development of genetic tests. So we have recently relocated to a new building in Kota Damansara that will house all the staff and departments under one roof. Of the company history. So MGRC was founded in 2004 and was awarded Bionexus status in 2007. The company initially focused on bioinformatics analysis and worked on several projects. So one of it being the genome assembly of oil palm that was done in collaboration with Sime Darby. In 2010, the company was listed on Bursa, and the following year we launched Detect, which is our genetic screening service. And I'll be talking more in detail about this later in the slide. In the following years, we have also been active in several sequencing projects with government and private sectors. Recently, since the last quarter of last year, we have ventured into cell immunotherapy. So these are our key management, with Dato Elvin and Sasha playing an active role in managing the projects undertaken by the company. So MGRC has two main laboratories at the moment. The first one is the Genomics lab, which is headed by myself, and the other lab is the cell laboratory, which is headed by Dr. Jolene. Since that this talk will focus on the genomics aspect of the company, I will briefly explain on the work that is done at the cell lab so that you have an overview of what happens. So the cell laboratory focuses on immunotherapy, which is said to be beneficial for late cancer patients. This is done by utilizing and enhancing a patient's own immune system ability to identify, target, restrict, and eliminate cancer, which will result in better survival rate, improved life expectancy, fewer side effects, and increased quality of life for the patients. So these are some of our partners and clients that we've worked with all around the world. So now I will go in depth about what we do in our genomics laboratory. So I'm assuming most of you who are listening are from the genetics club, so you should be very familiar with the basics of genetics. As you know, all living beings have DNA, the genetic code that encodes for RNA and protein, which eventually determines most of our phenotypes. So in genomics lab, we focus on the genome of any living being. So this includes things like, it can be humans, animals, bacteria, plants, and everything else in between. In our lab, we work with both DNA and RNA. So forgetting about any external factors such as environment and diet that can also affect your phenotype, the DNA within us will play a role in many aspects. For example, in determining our risk for obesity. So you may have noticed maybe some of your friends who eat a lot but they don't gain weight. So this is because of your genes. There's the same thing for endurance. Maybe someone has better endurance, so that would mean they would be more suitable for certain sports. For example, like marathon. So within the genomics lab, we do have another two divisions. The first one is for genetic screening, which we call as GSS. And the second one is for sequencing, which we call as GSA. So in GSS, the genetic screening, we look for markers associated with diseases, health traits, and reactions to drugs. So our company our company currently markets this screening test as DETECT. So the sample type that we receive are either human blood or buccal swab. In the sequencing division, which is the GSA, the focus is more on next generation sequencing and bioinformatics analysis. So this can be for whole genome sequencing, transcriptome sequencing, and also metagenomics. In terms of the sample type, since that we're looking at genome, it can be sourced from anything as long as it's a living being. So I'll go through in detail about each of the division in the following slides. So in the genetic screening division, we focus on parts of the genome that are associated with disease and health traits. Each one of us would have varying genetic material that would account for 0.1% of our DNA. So these small changes are known as SNPs, or it's called single nucleotide polymorphism, and they can act as markers that allow us to differentiate between individuals or species. In our DETECT product, we have markers that look at these SNPs. Through a process called genotyping, we are able to look at these changes and then tie it back to the risks associated with disease or health traits. The result that we get from this are very different than those of diagnostic tests. So this is the typical blood test that you go to a clinic to do. So it's very different than that. DETECT is about preventive healthcare. So with the information that you get from your genome, what you can do is make lifestyle changes to mitigate the risk. So I'd like to share with you a video that was done in collaboration with one of our partners that talks about DETECT and how it has helped one of our patients. So I think this would be a useful introduction. So I'll play this video.
doctor? Could we maybe stop the video for a while? Yes. To the guests, we shall take maybe a 1 minute break to get the audio back. So give us a minute, yeah.
Okay, sure.
Doctor, we can't hear the audio.
You can't hear the audio right now?
Yeah, throughout the whole video, we couldn't really hear the audio.
Oh, okay.
So share the macam mana Julia? Like you have to share the audio, is it?
Doctor have to stop sharing first, and while you are sharing the content, you have to include our computer audio. There's a button, yeah.
Okay, sorry everyone about the slight technical issue just now. We have corrected the audio, so I'll pass it back to Dr. Arshana now.
So I'll just show you the video that we've done with our partner, and it gives you an overview of what DTAC is about. Genomics provides a range of genetic tests to screen your DNA for disease and disorder risk, such as cardiovascular disease and cancer risk. With Genomics, you can also screen related to your overall health and wellness. Genomics test analyzes DNA to detect genetic variations known as genetic markers. Different genetic markers indicate different health risks. In addition, genetic markers may also indicate your risk for adverse reaction towards certain type of medications. With these results, you can now take action to mitigate your health risk. The process in the laboratory starts by extracting DNA from buccal swabs. Once the DNA is extracted, we will perform QC to check that the quality passes our requirements. This is followed by multiple steps of processing the DNA, starting from DNA amplification, fragmentation, and staining until we get data. This data is then processed into a readable report format that is easy to understand.
I wanted to do genetic tests because after I had cancer, colorectal cancer, I was looking for this specific test called BRCA. BRCA tests for Lynch syndrome, basically, the other cancers that are associated with colorectal cancer. And that was the reason why I was searching high and low in Malaysia for found it in Genomics. First, I found out that I do not have the genes for BRCA, which was a huge relief for me. And the other thing that I actually did was the oncology profile. So after doing that, I know I'm at higher risk for certain types of cancers, and it actually runs in my genes. And type 2 diabetes runs in my mom's side of the family, and I have high risk for that. So that told me the answers that I needed. Yes, I'll definitely apply it to my daily life because now I know what type of tests I should take in the future and to be regular about that. And the other thing is that I found out I have a higher risk for hypertension. So I have to be really careful, not too much strenuous physical activities, but I have to keep a healthy weight. So I'll definitely exercise regularly. And I did another profile to find out what type of deficiencies I have in terms of aesthetics and also my cardio and metabolism. So finding that out actually helps me know what type of supplements to take. So I'm definitely going to increase on my omega-3 oils, and I like vitamin D, so I'm going to increase on that as well. To increase green and leafy vegetables and also citrus fruits in my diet. I trust the Genomics test because it's established and it's under the Malaysia Genomics Research Centre. I would definitely encourage people to go for a genetic test because if certain types of cancer or even diabetes or hypertension runs in your family, you should find out. Prevention is always better than cure.
So that was a brief introduction, which I think will be more interesting than just watching slides. So we do have a wide range of products under Detect, but I'd like to introduce these 4 main tests that we have, which are our best sellers basically. So the first one is Detect Cardio and Metabolic. As the name suggests, this looks for cardiovascular disease and metabolic disorders. So this would be, for example, like stroke and gout. So another popular test is Detect Onco that looks at cancer, so such as lung cancer and liver cancer. We also have Detect Wellness that looks at general health traits such as obesity and stamina. In Detect PGX, this looks at a person's reaction towards drugs, and this information is very useful for doctors treating patients so that they can administer the correct dose of drugs. We do have a lot of other tests that we also sell. So some of our newer tests that we have includes the Detect Carrier and Detect Immune Health. So every year what we try to do is to come up with new products that we feel would cater to the current situation and be appealing to more people. And we also take in requests for custom tests. So recently we did have quite a few inquiries for neurology. So when more inquiries come in for a very specific test, we would then introduce it as part of our lineup as well. So in our lab, Detect samples are processed on a daily basis. The most common type of samples that we receive are blood and buccal swabs. We will perform DNA extraction using our optimized method and QC the extracted DNA. DNA that passes our QC requirement would then move on to our microarray chip. The data would then be processed using our own pipeline to generate the Detect report that analyzes all the huge amount of data that's generated. In the genomics field, relating to human disease, a background in medicine is also relevant because you could either be a medical officer or a geneticist that advises patients on what to do with your genetic results. So a daily life at MGRC's Genomic Laboratory would look something like this. Currently, our main focus is on the Detect test. So our team processes blood and buccal swab samples frequently, starting from DNA extraction to all the way to the data output. There would be optimizations carried out whenever we bring in new tests to offer or when we try new commercial kits. So whenever there are any research-based projects, the task would range from performing sequencing to something entirely new to the team, such as ELISA. Apart from doing wet lab, we would also need to do other tasks that maybe some would find a bit boring, but these are all important things to do. This would include general housekeeping, such as maintaining inventory to ensure there is no disruption of your work and deadlines can be met. So it's a horrible situation to be in when you have a tight deadline to meet and then you run out of reagents. So it's always important to keep your inventory in check. And then there is also, you might have to arrange for equipment maintenance so that everything is working in good condition. So most companies would also have their own internal documentation, such as standard operating procedures. In our lab, we also have working instruction, which is a document that specifies all our processes in great detail. So both these documents must be updated regularly. So it reflects current practices. So if a company is going for ISO certification, then there are more documents to prepare. Since we also take in research projects of transferring any pathogens to the team members. We would also routinely use glassware and sometimes blade. So this would pose risk. So it's important to always wear proper protective gear and dispose the sharp items properly into the sharp bin. We do use a lot of equipment in the lab, and some equipment would have different voltage requirement. So it's important to be aware of this and make sure proper precautions are taken. So when you're using certain equipment such as autoclave, it's very important to know how to use it and how to operate it safely because it uses high temperature. And another very common lab equipment is centrifuge that everyone uses. It needs to be properly balanced so that it don't damage the equipment and it doesn't pose a risk to yourself as well. And long hours in the lab would also take a toll on your posture. So in our lab, it's not a general practice to be standing throughout the whole process. So we do sit down, so it's not so severe. But in certain labs like pathology, most of the technicians would be standing. So that would, be like an ergonomic hazard. But in our lab, we do handle a lot of samples. So we do get sore hands, you see, from handling the pipette for the whole day. So this we sort of reduce and the discomfort by using multi-channel pipette or electronic pipettes. These are some of the challenges that we face in our job that I believe would be similar to other companies. So since we are a service provider, fast turnaround time is very important. So we would need to always meet tight deadlines. Most of the time, it would require you to multitask and prioritize certain tasks over others. This is something hard to do early on as you start to work, but with more experience, you will tend to get better at it. Being in this industry would also mean that you need to keep abreast about latest events and see how they can be incorporated into the company's line of service. So for example, recently we are trying to introduce a new test that looks at oxidative stress in patients. So we need to look up available products on the market that we can integrate into our service. So this would include, having to go through several literature, maybe engage with product specialists and test out these different kits, and also, performing new procedures that maybe we have not done before. So lastly, like any other workplace, there will always be a tendency where you will be assigned tasks that are not originally listed in your job description. So some people would not like this, but my personal opinion is that you should definitely try to learn as much as possible at your workplace, because this task might not be relevant to your job scope, but it can contribute to helping you improve your soft skill instead. So the very last part of my talk today will be on interviews, as I'm currently actually going through several applications to choose candidates to join our team. So I thought it would be something nice to add on and hopefully you would find it useful. So first of all, when you're updating your resume, please include only relevant information. It's not necessary to include things like your UPSR results. It's also important to practice to it's a good practice to customize your resume for each job application so that you can highlight certain skills that are relevant to the position you're applying for. So a common mistake that some of the fresh graduates tend to do is to copy the soft skills section of the internet. So whenever I question them about it, they can't answer. So it's very important to know what you put into your resume so that when you are asked during an interview, you are able to answer them. So providing a cover letter is also a great way to stand out from the other applicants. So cover letters are not like a written summary. It's not an essay of your resume. You see, it should be including, you should be including things that are not in your resume, but you find critical to share with your future employer. And whenever the interviewers ask about yourself, like ask you for an introduction, they're not asking how many siblings you have or where you live. What they want to know is, what you should be doing is take this opportunity to explain, like give a very brief introduction on your experience and what makes you a good fit for the position. So being a fresh graduate, you would also tend to feel that you don't have much working experience. I don't think this is an issue, because you should be using this chance to focus more on your soft skills instead and talk maybe about events that you have handled as a university student. So the employer can see how you will be a great addition to the team. So with that, I would like to end my talk. So if you'd like to know more about what MGRC does, or if you'd like to get in touch with us, you can always check the links that I've put up here. So I hope that this talk has been useful and has given you a glimpse about having a career in genomics. Thank you.
All right. Thank you, Dr. Arshana, for that very informative sharing session. I'm sure all of the participants would have found that information very beneficial. So for now, we shall be moving on to the Q&A session. But before that, for the ones who have just entered, I shall explain briefly about the flow of the event once again. Since we have finished the first slot, which is the talk given by our guest speaker, it shall be followed with a Q&A session afterwards. And also, like right now, you can just write out any questions that you feel relevant in the comment section to be conveyed to the speaker. And also, please do wait until the end of the event for a QR code to be displayed. Once you've scanned the QR code, it will take you to a Google Forms for the attendance and for feedback. Only the ones who have filled out this form will receive e-certs. So for now, we shall be compiling the questions and we shall go to the Q&A session. So before that, we shall take maybe a 2 to 3 minute break and we shall be back soon. Thank you. welcome back everyone. So right now we shall be moving on to the question and answer session. So we have received a few questions here. So let me start by asking Dr. Arshana, what advice would you give an aspiring student from the science background?
To enter into a genomics field.
It's a general question. It is as a graduate, what would you give them as advice?
Well, to be, I guess, to be practical, it's most of us would, as a student myself last time, of course, all of us would want to work in a field that is relevant to what we have studied.
Yes.
But I think it's quite difficult. Not everyone is lucky enough to do that. So I think like when you're applying for jobs, so just keep an open mind. So sometimes you might not necessarily end up in the field that you have studied, but you could be, ending up in a whole different field, but maybe that's a new passion for you. It could be that. Yeah.
All right. So moving on. How are the job opportunities when it comes to this particular field? Are there many industries like this in Malaysia? And will there be more coming in the future?
There are a few companies similar to what we do. In terms of job opportunities, I don't think it's, there are. If you're looking for lab to work in a lab, there are, but it's limited. But for sales, if you're looking for sales and marketing job in the same field, there's always a lot of opening. Because I believe when it comes to sales and marketing, the turnaround in companies are actually quite high.
Mm-hmm.
So a lot of people, after they maybe meet their, like they have reached their quota or something, then they would move on to a different company. But in lab, I would say it's definitely limited. Mm-hmm. In terms of companies offering like similar things that we do, there is quite a few, but you just have to look really hard for it.
All right.
Yeah.
So I think this is a bit more of a personal question. Mm-hmm. How is your experience working in this field? Is this job satisfying both financially and emotionally?
I would say personally I'm satisfied with this job, because when I first started, actually I started off as a molecular biologist, so that was the entering position for everybody who was going to go into lab. So for me, I guess I was quite lucky because like when I was not like entirely satisfied with my job, I did speak to my management, and they did take corrective action. They did. They gave me more opportunities to learn new things and then to get more responsibilities. So like every few years, I've always had the chance to learn new things in my company. So it's not entirely routine. So in terms of job satisfaction, I would say I'm pretty happy. In terms of salary, I can't speak for everyone because it depends for each company. It depends on the position that you're working for, and also depends on how, like financially how well your company is doing. So if your company has lots of sales coming in, obviously you will be paid well. But if the sales are low, it's hard to expect a very high salary. It's hard to expect like an increment every year. So it's a trade-off.
All right. Mm-hmm. There's also a question asking, do you consider your job fun?
I would say fun, yeah. I mean, of course, there are some days where it's not fun. But overall, I think, yeah, it has been pretty good because, like I said, I always had opportunities to learn new things because it's also important for the company to be dynamic. You do not want to remain stagnant, because if you're ever stagnant, then like the plants that we wanted to test, we wanted to feed the compound to animal. So it's just like animal study. So we needed someone to have a background in that.
All right.
Yeah. So it's not something continuously that we do. It's just a one-off project that we had.
I see. All right. What are your thoughts, Doctor, on gene therapy?
Oh, gene therapy. So I think that one is more on like it's definitely not something MGRC does. But it's maybe it falls a little bit on the cell laboratory because you're looking at like cancer. Yeah. So are you referring to that?
Yes. Like, what are your thoughts on it? Should it be something that we should practice or should initiate here in Malaysia?
I think, yeah, why not? Because if it helps to, I mean, it depends on the purpose of your gene therapy. If you're looking at more on, overcoming illnesses, then it's fine. But gene therapy, sometimes I guess people kind of mistake it for, like choosing your eye color and things like that. So that is a no. I would say no. But if it's for disease and illness, it definitely helps. But it also comes with a high cost. So, for example, our cell therapy lab, it's a lot costlier than the service offered by that division is a lot costlier than our detect because of the process itself.
I see. All right. So now we have a question from Munira. What advice would you give to your younger self at the start of your career, and what impresses you the most when you are in charge of interviewing someone, especially for undergrads?
What I would tell myself. Well, I guess, I mean, in the beginning of my career, I did feel that having a PhD was not an advantage for me because I felt like the opportunities were quite limited. But I guess now it's a bit helpful because I've already had a few years of experience. So I would say, what would I advise myself? I guess be vocal. I think that's important because if like in the beginning of my career, if I never approached my management and said, I'm not happy with this and this and this, I would probably just be unhappy in my job anyway. So I think if there is certain things that you feel can be changed, you should try to be vocal and discuss with your superior and see if changes can be made. Maybe a compromise can be reached. That can be a win-win situation for both sides. So in terms of what was the second question about the interview?
What impresses you the most when you're in charge of interviewing someone, especially undergrads?
Okay, like impressive about the candidates, you mean?
Yes. What makes you interested in taking them, selecting them?
Okay, all right. For me, I guess if I do look at first of all, I definitely look at their background and to see it's understandable as a fresh graduate you wouldn't have much hands-on skills, so I understand that and I'm totally okay with that. But I look more on soft skills because for me, I think being a team player is really important. It's very hard to have lab members who do not want to cooperate and work together. So usually I will try to see if the candidates can work in a team or they would just go off and do their own little thing. So it's really hard to work with people like that. And also in terms of technical skills, I also look at if they can think, have critical thinking is really important because sometimes during the interview, some of the candidates come off as very technician-like. So they just do something routinely and they just do it without thinking. And it might work in certain field, like if you're in a pathology lab. But for me, I don't want that in my team because then I always have to spoon feed. I always have to give all the information. So it becomes tiring and it becomes pointless actually. So to hire someone like that. Yeah.
Right. Okay. Another question is, since basically if this field is progressing, then there's more genetic testing that's being conducted, especially on patients. So we have a question from Rishika. Do you think that parents deciding to do these tests on their kids at an early age would cause any fear for the kids' health? Thinking about the possible diseases that they could have, would they be living in fear their whole life? What would you advise them?
Okay. Well, to be honest, actually, so I have done the test myself. So I have the reports. So So when I first did it, I also, had this fear, like, oh my God, I'm going to get this cancer and things like that. But truth be told, this is just, like when we look at this the detect test just looks at one part of a factor that can contribute to a disease, which is your genes. But there's so many other factors. There's things like your diet, your environment, your lifestyle. So if you're maybe a smoker versus non-smoker, your risk for lung cancer is going to be totally different. So even though you might have the gene for it, but you can control certain parts of your lifestyle. So if children do it at a young age, there would be that fear, I guess, but it depends on. You really need to educate them to say that it's not like 100%, certainly you will die out of cancer kind of a situation. We do have that disclaimer on our report anyway, so you don't have to like be so fearful of it, I would say.
All right. Okay, I have a very good question here from Rishika.
Okay.
Since now, it has been almost a year and a half of the pandemic. So most of us, especially in the science background right now, all of our lab sessions are theoretical. All of our lab sessions are online. So we don't even have hands-on experience like micropipetting or doing this kind of stuff. So will that affect us when we come out later as graduates? Will that be a problem?
I think it's understandable because of the situation. But so if, like for me, if a candidate like that came, to an interview, what I would see more is like on their enthusiasm, because I've had like an intern before who, did not have much experience in lab, and during the interview he said he was eager, but when he joined the lab, he was not eager at all. So the opportunity was presented to him, but I feel like, it was a wasted opportunity. So definitely be enthusiastic, even if you do not have the hands-on experience. You should show the interest that you're willing to learn, that you are curious. And I think that would come off as a really positive trait during interviews.
So it's more to the candidates' characteristics and how they want to. Yes.
It's understandable, as a fresh graduate, you would have very limited, hands-on skills. So it's very understandable. So if an employer thinks otherwise, I think that's not fair. Yeah. So for me, I wouldn't be so judgmental on the technical skills if you're a fresh graduate. Yeah.
All right. So another question from Aisha. Do you think that the genomic industry will have a bright future in Malaysia, as this field is not very well known here?
I don't think. I'm not sure where the impression is coming from that it's not like well known, because we have been in this industry for a long time, since 2004, and we have been able to sustain this long. So I don't think it doesn't have a bright future. I do think there is a bright future, because there's always new technologies coming up. There's always new findings. So it's always it's a growing field, definitely.
All right. I think the impression is coming from the fact that there's not that many industries that give this. So another question is, how do people get tested genetically? Are they recommended by doctors, or can people do it anytime? Like if they decided that they wanted to do a genetic test, can they just walk in and do it?
Yes. So it's not recommended by the doctors usually, because these tests are more on looking at your risk. So it's completely different than diagnostic tests. So it just looks at, like what is the possibility of you getting a particular illness and things like that. So like for me, I would suggest someone to do it at a younger age, because if you know your risk of a particular disease at a younger age, you have much more time, to fix your habits, make lifestyle changes and things like that.
I see.
So if anyone who's interested to do the test, you could go to a clinic, but I believe not all clinics offer this. So if you're really interested, you can get in touch with us, and then we can connect you to the clinics.
I see.
Most of the time, we do not allow walk-in patients, because we do think it's good for the doctors to explain the results and guide the patients, walk through the whole thing with them.
All right. So what are the initiatives that are being taken in order to spread the importance of genetic testing or the or in the genomic industry to people without the science background. Like they don't really understand the importance of it or the significance of it. So what are the initiatives taken to strengthen that ideology?
Okay, so what MGRC does is usually we work with clinics, we work with doctors and hospitals. So most of the time, what we do is organize workshops for doctors and educate them actually. So then it will be the doctors, who would then continue the, passing on that information to their patients instead.
Right, I see. All right, I think we have reached the end of the questions. There's quite a lot of questions also. So okay. So with that, we have come to the end of the Q&A session. Now I shall invite the advisor of the Genetics Club of UKM, Dr. Izwan Barudin, to give some closing remarks. Dr. Izwan?
Okay, thank you, Hashwin. Okay, first of all, I would like to thank Dr. Ashana Amin for her time, precious time on weekend studies. And furthermore, I would like to express my appreciation to the all organizers, UKM, MJRC, PMFST, especially to the secretariat of the technical teams of the Genetics Club of UKM for their hard work in making sure that the success of this webinar today. And personally, I think this is a very good session between the industry and the students. And so all students who will be job seekers, I think, in the next few years must grab this opportunity to expand your knowledge and polish your skills needed in this field while you're still in UKM. And I think all students should take the note some of the important information mentioned by Dr. Ashana, such as the experience, the soft skills, the technical skills that you need to enter this field in the future. And I believe that all participants who are attending this webinar is also getting some general information regarding the prospect career in genomics basically in Malaysia, I think. And it is my great pleasure that we have a fruitful discussion, though I think 1 hour is not enough. So on behalf of Genetic Clubs of UKM, I would welcome the opportunity to return with another sharing session from other different group of speakers to talk on the future job prospect in genetics in the future. Okay, thank you. Thanks again, Dr. Ashana, for your informative talk today.
Thank you so much for having me.
All right. Thank you, Dr. Izwan, for your kind words. We have come to the end of today's webinar. I, as the moderator for this event, would like to thank Dr. Arshana for accepting our invitation to speak. Your input is greatly appreciated, and I'm sure all of the attendees benefited from it as well. All the participants are advised to wait for the QR code to be displayed. This QR code will lead you to a form of attendance and feedback. For those who do not have another device to scan with, the link to the Google form will be uploaded in the comment section as well. Thank you and have a nice day. Thank you again, Dr. Arshana.
Thank you.